Thank you very much, and good to see you again. I hope that after this we'll have more opportunity to interact. So, as Michael said, I'm the EMA support for Extractable and Leachable for our European experts. One is in quality, Catherine. She's the real expert, and I mean, the two of them are expert, and I'm just saying I'm doing the supporting work. And then there is Leon for non-clinical. So you're very lucky actually to have Catherine joining us because she's got a lot of hands-on experience on the discussion. I think actually she just come back from the ICH meeting, so she might be a bit jet-lagged. Okay, we get started again with Slido, now our best friend. It worked before. I'm going to see if the same number of people are falling asleep during this part of the training. And the first question is, what EU documents make reference to extractable and leachables? Notice to applicant, guideline on plastic immediate packaging material, none of the above, all of the above. I think numbers are stabilizing. Second question. Does complying with foodstuff waives requirement for extractable and leachable studies? Yes, no, only for solid dosage form, none of the above. Okay, you got them? All right. And next and last question. Does compliance with the chapters of the PHR for packaging material always waives the need to conduct extractable and leachable studies? Yes or no? And we are aiming again to 90% right answer by the end. So my part of the presentation focuses on plastic material. So we are going to look at the EU legislative framework and regulatory requirements for plastic packaging materials, starting with the notice to applicant, the guideline on plastic immediate packaging material, the EMA question and answer, although are not strictly related to extractable and leachable, the Pharmacopoeia European general chapters, and the food contact legislation. So the EU legislative framework, which is applicable for human and veterinary medicines, actually do not know whether maybe your agencies also cover veterinary medicine, and you are also maybe involved in the review of veterinary medicines. So there are some aspects that are mentioned in the directive. So in the directive is like really high level. requirements are briefly mentioned in Annex 1. But then a little bit more information is provided in notice to applicant and basically requirements for active substance, pharmaceutical development, and requirement for finished product. We'll have to have a look at this in more details in the next slide. The main guidance is the guideline on plastic and immediate packaging material, which is a QWP and also CVMP guidance document. We will look at food contact, PHUR, and as the general chapters, and as I mentioned, also at the question and answers. So notice to applicant. Okay, so the requirement for primary packaging is that material of which primary packaging component needs to be stated. And the specification need to include the description, identity, critical dimension drawings, and non-compendium methods with validation. So for the secondary packaging, if no additional protection is provided by the secondary packaging, the description is sufficient. But if they also are functional secondary packaging, their function is in addition to the protection, the notice to applicant says that additional information is required, but it doesn't state specifically what is required. Then the suitability of the packaging needs to be discussed with respect to choice of material, protection from moisture, light, compatibility, sorption, leaching, safety of the materials. Before, when we had the presentation on inhalation products, I had an inhaler. Now for extractable and leachables, I'm going to do this from my plastic bottle. So here you already have a part of the answer to question 1. Now let's move to guideline on plastic immediate packaging materials. And this is our main guidance in Europe to date. It refers only to plastic immediate packaging materials, elastomer, rubbers are out of scope. And it describes the minimum level of information required per finished product type and also in what location the information needs to be provided. So I think a key point to consider here is that is the minimum level. So if this information is not provided in the dossier, ask for it. And then, depending on the risk-based approach, even if ICHQ3 is not yet available, additional information can be asked. So it's also got a correlation table given for veterinary products, and the different requirements are described in different sub-paragraph. And in this presentation, for ease of reference, I maintain the same numbering, and I found this helpful, particularly when I gave a similar training to my colleagues in the quality office, because then it's handy to refer back during assessment. It makes references to other documents, legislation, the Pharmacopoeia, the European Pharmacopoeia, also Pharmacopoeia of member states, and the food contact legislation. So although this is a quality working party guidance, it actually focuses on safety and compatibility, so on non-clinical and clinical aspects. And protection and performance of the container are not covered in the guidance. They're covered, as we've seen, at a very high level in the notice to applicant. He applies a risk-based approach, and this is related to the physical state of the active substance and the finished product. So there are different requirements for solid versus non-solid, and based also on the route of administration. So if it is oral, ophthalmic, topical, parenteral, or by inhalation. And extractable and leachable are not covered as extractable and leachables per se, but are included under extraction and migration studies. And there is no specific guidance on how to evaluate them. So that's on the methodology. So that's a limitation of the current applicable guidance. Here, I'm not going to go through the details on the table here yet, because we will cover them when we get through the requirements. But basically, just for ease of reference, I divided what the requirements are based on the guideline layout, so 3.1, 3.2, 4, 5, and 6. And what the requirements are and where they should be presented. So under S6 for the active substance, or in between P7 and P24 for the finished product. And then we will see what these requirements are in the following slides. So, extraction studies. They aim to determine additives that might be extracted. They are required for plastic materials of non-solid active substances and finished product. For products that are oral and topical, non-ophthalmic, if the Pharmacopoeia Europea or the Pharmacopoeia of a member state it does not include them, or if they're not approved for foodstuff packaging. For non-compendial materials for inhalation, parenteral, and ophthalmic use, even if approved for food packaging. And then we look at the decision tree, which I think is actually quite helpful. Then the testing involves exposing a sample of the component to an appropriate solvent system under stress condition in order to maximize the amount of extractables from the packaging in the solvent. The solvent has the same extraction potential of the active product. This is a requirement. It would be beneficial if the solvent had the same extracting potential of the active substance or the finished product. And for the finished product, the ideal solvent is actually the product per se or placebo and vehicle. Then the specification of the packaging material need to state the nature and amount of the extractable. The interaction studies, so the aim is the compatibility with the selected plastic packaging material under the intended use. And there are two types of studies that are done during development. One is migration, and migration is basically the release of the substances or the leachables from the plastic component into the content of the container under conditions which reproduce those of the intended use. And they're necessary when leachables have been resulted from extraction studies, unless the maximum level are regarded as safe. Sorption, these are the development studies to investigate the sorption of the active substance of an excipient to the packaging material. And these are generally performed if changes are detected during the preliminary stability studies. So migration are leachable, but sorption actually does not fall under extractable and leachables. It is the inverse kind of phenomenon. Testing might be performed by use of plastic material or plastic component of the container itself. And the extent of the design of the interaction study depends on the physical state of the active substance and the dosage form of the packaging product. So they are required for all non-solid active substance and finished product, even if compliant with the PHUR of the food stuff. So pay attention just in case for the question. And they may be required for solid dosage form for inhalation and parenteral use. And we said that actually it's a safety guidance with the safety consideration. So toxicological data should be provided for extractable and leachables depending on the level and chemical structure. They're not needed if the additives are used as prescribed in the Pharmacopoeia Europea or the Pharmacopoeia of the member state for a given plastic material container for a given use. So although there could be compliance with the European Pharmacopoeia, it is important to check that is that specific plastic material or container, and that it is for that use, for that purpose. Plastic or additives that are approved for use in food packaging, also the data doesn't need to be provided unless it's used in packaging of the finished product for inhalation, parenteral, and ophthalmic use, because these are more risky routes of administration in terms of the impact of extractable and leachables to the patient. So I personally find the decision tree, this flow chart, really helpful, because anytime I start reading the full text, which I try to summarize, then I get a little bit lost and I keep on having to go back and read again. But the flow chart for me is very helpful. And so there are two of them. One is for active substances. So you see that for solid active substances, we don't need to request much information for the packaging material. So we need general information and specification. So basically, we need the chemical name and material and any monomer used, and then either an in-house monograph and a certificate of analysis with the description, identification, and properties, mechanical and physical. The requirements are different for non-solid active substances. So if the material is described in the European Pharmacopoeia or in the Pharmacopoeia of a member state, and in accordance with this foodstuff, then what we need is, again, general information, specification, and migration studies. But if the material is not compliant with the Pharmacopoeia or with the foodstuff, then in addition to the general information, the specification, the migration study, we also need extraction studies and toxicological documentation. Things get a little bit more complicated for finished products because now there is not just a differentiation between solid and non-solid, but also the route of administration. So those routes of administration that are considered less risky or of exposure of extractable and leachable for the patient, the requirements are very similar to the requirement of the active substance, basically, in terms of level of information. But if the requirements, if the route of administration is inhalation, parenteral, or ophthalmic administration, then first of all, the compliance with the foodstuff legislation is no longer taken into account as a justification for the provision of extraction studies and toxicological data. So the compliance with foodstuff is no longer sufficient. And then you need to be aware that when there is compliance with the European Pharmacopoeia or a Pharmacopoeia of a member state, it's important to look at the monograph, because if the monograph authorizes the use of several additives from which the manufacturer might choose one or several, then we need the compliance with the Pharmacopoeia is no longer adequate. It needs to be is no longer sufficient. We need to have extraction studies and, if needed, toxicological information. So now looking at the question and answers for packaging published in the EMA website. So the first question is highly relevant to what we just seen so far, to the guidance. and is related to the acceptable quality standard for plastic materials for containers in solid active substances and solid dosage form. And actually, we do get as a question or even during assessment, what happens if they're not compliant with the Pharmacopoeia? Do they have to comply or not? So reference to specification published in the Pharmacopoeia is not always possible and is not obligatory, as the chapters describing the material are not exhaustive in the Pharmacopoeia. And plastic materials compliant with the foodstuff legislation are acceptable. And a specification should be presented in accordance with the guideline on plastic and immediate packaging material. So actually we see that this question and answer amends to a certain extent the requirements the requirements that we saw in the guidance, because in the guidance, if it is a solid dosage form, we need basically a description and a specification. Here, and it doesn't say that they need to comply with the Pharmacopoeia or the foodstuff, but in this case, the question and answer from a quality perspective gives additional reassurance that compliance with foodstuff has a value in the assessment of the packaging material. Then the second question and answer is related to aseptic processing in your terminal sterilization. And we do get actually this point quite a lot during assessment, and it can lead to major objection because for a finished product that they've been manufactured in heat labile packaging material, the applicant is very reluctant to change the packaging and to change the sterilization method when we actually point out that we need information to demonstrate that the finished product cannot be thermally sterilized. So we give precedence to terminal sterilization over the choice of heat liable packaging material. So if a finished product, be it eye drops or a sterile solution, is provided in a plastic container, which is heat labile, and then the applicant basically justifies the choice of the sterilization method based on the container, the answer is that it cannot be accepted. So aseptic processing, it cannot be considered to replace the same level of reassurance of sterility than terminal sterilization. And that is particularly for products that have been developed in America and then they try to come to the European market. It's quite a sticky situation because they say, well, it's approved by FDA and why won't you accept it? And the reason is because of the level of reassurance of the terminal sterilization method is much higher, and if you look at the guidance, there are several steps. The guidance on sterilization, there are several steps that the applicant needs to go through to demonstrate that sterile filtration is actually the only option that they have. It has to be based on the finished product, not on the container. But what about the data for the sterilization process of the primary packaging material used in aseptic manufacturing? So if at the end the applicant justify the use of aseptic manufacturing, what do we need to see? And sometimes this is actually not always assessed in such a detail. So it's good to keep in mind that we need to have the primary packaging material sterilization method and cycle, because it's a critical process and sterility is considered a critical quality attribute. We also need to see the validation of the sterilization cycle if the conditions are not stated in the Pharmacopoeia. And the name and address of the sterilization site and GMP certification. And here we say when available, and then it gets a little bit more complicated depending if the packaging material is also a medical device. So for CE mark class 1S medical devices, so sterile syringes, a declaration from the manufacturer that the component is CE marked, the sterile device, together with a copy of the certificate of conformity from the notified body needs to be provided. But in the absence of the GMP certification of declaration that the component is CE marked, then we need a confirmation that the sterilization process has been conducted and validated in accordance with the relevant ISO standard, which is ISO 6 ISO listed in the EMA website. And this point, they sometimes get missed out, so it's good to keep them in mind. And the site where the QP certification of the finished product takes place and the site responsible for outsourcing the sterilization activity should have access to the necessary information to demonstrate that the ongoing qualification status of the supplier of the sterilization service is available, because this can be checked during inspection. So it's a good point to double check. The responsibility for the sterility of the packaging and the finished product manufacturer, and he also incorporated in the sterilization guidance. This information is incorporated in the sterilization guidance. Then let's have a look at the requirements in the European Pharmacopoeia. So I'm sure you know that the European Pharmacopoeia is published by EDQM. Plastic materials, plastic container enclosure activities under the responsibility of Group 16. And the European Pharmacopoeia is legally binding in Europe. So if you're familiar with the Pharmacopoeia, you know that there are 3 sub-chapters. So there is a section 3 that is dedicated to containers. And this has 3 sub-chapters. So the first sub-chapter is materials used for the manufacture of containers. And the second sub-chapter is actually containers, and it gives example, different type of containers. These sub-chapters are applicable by reference. So when they are referenced into the pharmaceutical preparation monograph and to the dosage form, monographs, then they become mandatory. Then in 2017, EDQM had a survey because there was a need to update, to take into consideration the medical device that are also container closure system. So then a third subchapter was created that has come into force with the 10th edition. So these are medical devices and the materials of manufacture, and they are located in subsection 3.3. And similarly to 3.1 and 3.2, 334 is referenced in the anticoagulants and preservative solution for human blood. So then it becomes mandatory, and is also referenced in human plasma for fraction and human plasma pooled and treated for virus inactivation. So unless referenced, these chapters are for information, but if referenced, they are mandatory and they need to be implemented. And compliance needs to be demonstrated in the module 3. Group 16 actually has been very active lately. Here I've summarized only some of their activities that are ongoing. So for plastic containers, well, they stated that all containers, they have these extractable elements. So all containers for pharmaceutical use, described in chapter 3.2, are components of a medicinal product, and because of this, they're subject to ICHQ3D for elemental impurities. Heavy metal test has hence been deleted from rubber closure for container for aqua preparation and for powders and free-dried powders. For the plastic material for containers for pharmaceutical use, there is a proposal to replace the heavy metal test with ICP-MS, which is in line with guidance. And a separate general method for determination of extractable elements in plastic material for pharmaceutical use has been elaborated. The draft test 2435 was published in Pharma Europa, and comments are under consideration. So perhaps if you work in this sector, you might want to consider sending comments, unless they're closed already. But it's interesting to see the direction of travel that is being taken by Group 16. And these are other texts that are underway. Yeah, so with the reference to the Pharma Europa, where they've been published, there are cycloolefin polymers, cycloolefin copolymers, styrene block copolymers, etc. So several other chapters are under revision, and they're available for information under the knowledge database. And last point for consideration is suitability for food contact that we see, according to guidance, sometimes can replace the extractable studies. So the framework is the regulation 1934-2004, and basically states that the food contact material shall be safe and not transfer the components into food in quantity that could endanger human health, change the composition of the food in an unacceptable way, or deteriorate the taste and odor of foodstuff. It sets legal requirement on chemical migration. There are some specific measures for a number of material, including plastic, adhesive, coating, and printing inks, and out of scope is glass and rubber. So here there is a comparison between the food legislation and the medicine legislation. In the food legislation, there is a positive list of permitted additives and monomers. So only those that are in the list are acceptable, while for medicine is a free choice, but the composition of some material is given in 3.1 of the European Pharmacopoeia. The use of new materials after toxicological evaluation and inclusion in the positive list is possible. While for medicine, new materials are evaluated during the marketing authorization review. And migration for the food legislation, migration limits set for individual monomers and additives are given. While for the medicines legislation, the reference is that there is no impact on safety, efficacy, and quality. So it's a different type of approach. Tests for compliance with migration limits using simulant under specified test condition is how the tests are performed, while in the medicine legislation, extraction study to screen for potential leachables is done, and then a migration study with a drug product solution. Migration substances that migrate up to the specific migration limit are tolerated if the taste or odor is not affected. So then the conclusion is that the material is suitable for packaging of food. While for medicine legislation, even if below the toxicological limit, the limit is not acceptable in case of changes to the finished product quality. And then at the end of the evaluation is the compatibility of the finished product and primary packaging has been demonstrated. We did receive a question about what information needs to be provided. So I summarized them in the module 3. I summarized the relevant information and the source of the documentation as per guidance in this table. And here are the requirements also for sterile products in terms of the stabilization of the container closure system, just as a point of reference that might be helpful during assessment. Okay, let's see how this time the presentation went. Personally, I don't know, like I find the extractable and leachables a little bit more drier than than inhalation products. But let's see if I manage to convey a little bit of the information. So what EU documents make reference to extractable and leachables? Is it the notice to applicant, the guideline on plastic immediate packaging material, none of above, all of the above? Very good. I'm pleased. Yeah, and actually, well, maybe, I don't know if in this instance more people were asleep, it looks like, but it's understandable. Next question. Does complying with food stuff waive the requirement for ENL studies, acceptable and leachable studies? Yes, no, only for solid or dosage forms, none of the above. And this is a bit of a tricky question. All right, I'm here. I'm happy that people are discussing, at least that. But well, I need to change my presentation skills or update them because depending, if we look at the guidance document, it would be none of the above because actually for solid oral dosage form, we do not need them, but if we look at the question and answer, then for solid dosage forms and active substances, then it does waive. It got a little bit better, but not so much. Okay, next time I need to think about something different. Next slide, I take suggestions. Right. And last question, does compliance with the Pharmacopoeia always waives the need? This is a tricky one. Always waives the need to conduct extractable and leachable studies. Good, so I was more effective on this part of the presentation. It is correct that is no, because it depends on the components that are listed in the Pharmacopoeia European. If there are more than one additives, then the studies needs to be done. Okay, well, thank you very much. And now I give the floor to Catherine. Catherine, are you jet lagged? Are you okay?
Both. Yeah, I'm okay, and yeah, I slept 3 hours last night, but I think this should work at least for the next few minutes or half an hour or how long it will take. No problem.
Thank you for joining. Thank you.
Yeah, thank you for asking me to present here. So I would like to illustrate a bit more what is expected in the actual submissions on extractables and leachables. On the next slide, I have a short definition again of extractables and leachables. Can I move the slides myself? Oh, no, sorry. Okay, so these are the key definitions. An extractable is a chemical species that can be released from a pharmaceutical packaging or delivery system, or packaging component or also from single-use manufacturing equipment under laboratory conditions which are somehow exaggerated compared to the final use of the component. So that means that specific extraction solvents can be used, and also elevated temperatures, and to shorten the duration of the study. And extractables are those substances contained in a component that have the potential to leach into a drug product formulation under normal conditions of storage and use. So the number of extractables is higher than will be in the end translated into leachables. And leachables are a chemical species that migrate from a pharmaceutical packaging or delivery system or component or whatever component in contact with the drug product under the normal conditions of use or during accelerated drug product stability studies. Leachables are typically a subset of the extractables or are derived from extractables, and that's why in this illustration here in the middle, leachables are not perfectly fitting in the extractables circle, but also a bit outside of the circle, because there may also be the situation that a component migrates into the drug product and is further degraded over time. So it's not one-to-one that leachables are only a subset of extractables, but can also be different. But the main number of leachables will be a subset of the extractables. Next slide, please. So the difference is the potential impact on patient safety. And the impact needs to be evaluated. So an extractable has a possible impact because it's not for sure that it will occur as a leachable, but the leachables are those substances which actually may have an impact on the patient's safety or on the quality of the product, and by this on the patient safety. And the testing for extractables aims to test the component or the material for a component selection. It's during the development process, and leachable testing is done in the final drug product. Next slide, please. So this is a rough overview of the analytical techniques which are used typically for analysis of extractables. So there is a lot to cover: anions, cations, volatile, semi-volatile organic compounds, and non-volatile organic compounds. Also silicon oil may play a role because many primary packaging components are siliconized to improve machinability, and silicon oil may also occur as a leachable in the final drug product. And a variety of analytical techniques is used to address the extractables in the respective study, and has to be used for screening. So the requirements regarding screening methods are they can only be semi-quantitative because you may not be able to really validate them, because you don't know what you're exactly looking for, and therefore you have to ensure that the methods are qualified, but validation is not expected. Formal validation, according to GMP, is not expected at all for such type of material characterization studies. Formal validation is another issue which is handled under GMP for the QC methods. But this is development studies, and so in that context we talk about characterization, and a formal validation is not required, but nevertheless the analytical procedures need to be reliable, and this should be ensured. Next slide, please. So how to approach exactly an extractable leachable evaluation. So first of all, or first step is the execution of an extractable study on, for example, the primary packaging component, and what has to be considered for that. As Claudia already mentioned, the components should be pre-treated according to the intended pretreatment for the commercial process. For example, if you want to investigate a rubber stopper, or rubber stopper that is usually pre-sterilized or can be pre-sterilized, and then depending on what method is used for sterilization, irradiation, or steam sterilization, the component should be pre-treated as accordingly. And also the other use conditions which are meaningful for the actual use. When you have done this, you do your extractable study with representative extractive conditions. That means that you don't have to fully degrade your material. You don't have to do a fully exhaustive study, so that in the end perhaps your component is completely diluted. But it should be exaggerated compared to the actual intended use. Otherwise, it wouldn't be an extractable study. And after the extraction, in the ideal situation, the compounds that have come out of the component are identified and can then be targeted in a following leachable study. And then the toxicological evaluation of the leachables should be executed as appropriate. So if you, for example, have let's say 5 or 6 extractables identified, then you follow them in a leachable study, and in many cases those leachables are at much lower levels than the extractable. So perhaps this alleviates also the toxicological evaluation. I will come to this later. A possible way to approach the tox assessment is described in a publication of PQRI on OINDP, on oral and inhaled nasal drug products. So there the concept of the analytical evaluation threshold is used for the toxicological assessment of the leachables, and the analytical evaluation threshold is based on the safety concern threshold. And I put the reference here below in the slide. You can read this in this publication. On the next slide, I will further explain. So the safety concern threshold defines the dose below which a leachable would have negligible safety concern, or present a negligible safety concern from a carcinogenic and non-carcinogenic toxic effects. So a safety concern threshold is proposed in the PQRI paper on OINDPs of 0.15 microgram per day. This is true for the class 3 Kramer class 3 compounds. These are the compounds which are known to or expected to have a genotoxic or carcinogenic effect. And this safety concern threshold can be used to calculate the analytical evaluation threshold based on the allowed daily dose. So you know how your product will be dosed, how large the volume will be that is applied, or whatever kind of dose. And this means, or this can then be translated into the AET, or this knowledge can be used to translate SCT to AET. That means that the AET is a product-specific calculation, and it's different for each and every product. Next slide, please. Aspects of the SCT are also described in the ICH M7 guideline on mutagenic and carcinogenic compounds. And the SCT is dependent on the daily dose and on the duration of treatment, and also on the route of administration. So this is an example here where it's described for the different durations of treatment, and you can see if it's used for chronic treatment for more than 10 years to lifetime, the daily intake may not exceed 1.5 microgram per day. Next slide, please. Here it's illustrated. I took this slide from a PKI presentation, which is also available in the internet, and I put also the respective link below for your reference. So this is just to illustrate the concept of the AET and how this strongly reduces the work to be done by a toxicologist for risk assessment. So if you calculate your AET based on your knowledge on the daily dose and the SCT, you see that here in this chromatogram, many peaks occur, but only 1 to 4 peaks occur above the AET, and only for those a toxicological assessment is needed. And of course, toxicological assessment is preceded by identification of the substance, or at least some structural information about the substance. So this is needed by the toxicologist to do a proper assessment. Next slide, please. So then in the leachable studies, you target the potential leachables that were identified in the extractable study. And it's again executed with components that have been pre-treated, like foreseen in the commercial manufacturing process, for example, irradiation or steam sterilization. And leachables are investigated in stability studies with a drug product or a simulation solution under accelerated conditions. I would say and simulation solution under accelerated conditions, but this depends on the nature of the drug product. We will see this in the last slides. The leachables assessment is executed during the pharmaceutical development. That means that the data are expected in the respective part of the CTD, meaning in P2. The leachable studies should be executed with the final drug product, if possible, and leachables should be monitored during the stability studies that are used for the application, for the exhibition batches usually. Accelerated conditions may be used in addition, and where it makes sense, and these are also very valuable information, especially when it comes to chemically defined drug products. There you have or very often the situation that upon submission, only limited stability data are available, let's say 12 months under real-time storage conditions and 6 months under accelerated storage conditions, and you can use these data to justify or to investigate the leachables and to conclude on safety of the or whether or not additional information is needed. Next slide, please. So there may be the situation where leachable study is not possible in the final drug product. For example, when you have polysorbate in your formulation, I took this these both chromatograms on the right-hand side of the slide from a publication made by colleagues here in the bee farm. The lower part of the figure B shows the chromatogram of polysorbate containing drug product solution, or it's a placebo solution with polysorbate. And you see there are already a number of peaks. And the leachables are or can be seen in the upper chromatogram called A. And yeah, it's quite difficult, or can be quite difficult, to identify leachables in the presence of polysorbate. And in such situations, it's justifiable to use a simulation solution to do the leachable studies, because leachables can be yeah, better investigated and yeah, identified, analyzed when you don't have that interference by polysorbate. And of course, the simulation studies, I think it's on the next slide. Yeah, simulation studies or simulation solutions should be selected according to the leaching propensity of the actual drug product. And yeah, this is achieved by mixing organic solvents and water, usually, yeah, taking into consideration the polarity of the mixture and the pH value and so on. And simulation studies may be executed under accelerated conditions, because leaching from components is following or is considered following the Arrhenius rules. And so that at higher temperatures, leaching occurs more quickly. And simulation studies are also often used to qualify manufacturing equipment, single-use manufacturing equipment, like sterile filters or yeah, bags used for buffer preparation or something like things like that. The sterile filters manufacturers often have, I would say, generic extractable and leachable studies at hand that they can provide using a variety of solvents, aqueous solutions at various pH values, and also mixtures of organic solvents and water. And yeah, when these studies are properly yeah, represent the actual in-use conditions, it's especially for manufacturing equipment usually acceptable to use those studies if the compounds coming out of the material are all below the toxicological limit of concern. So I think this was my last slide. Okay, yeah. No, I'm sorry. I wanted to talk about Q3E as well. Sorry. It's just that the next slides won't be so detailed and concrete as the last ones, because I can at the current stage of development of the guideline, I can only talk about the concept paper, which is already public. So the Q3E guideline is intended to complete or to further expand on the guideline family Q3, which addresses the several varieties of impurities in drug substances and drug products. So in Q3C, we have the residual solvents addressed, and Q3D, the elemental impurities, and Q3E, the extractables and leachables. Next slide, please. So the aim of the Q3E is to harmonize the expectations on extractables and leachables assessment, because the topic is out there, and we as agencies, we usually receive data because industry knows that there's something to be done. But the guidelines or the guidances or the expectations across regions are not yet harmonized. So this is in the end the aim to help on the one hand the companies to get assurance what is actually expected, and also to guide assessment of those data. And so the guideline intends to provide guidance on the conduct of the extractable leachable studies. This is more the quality, the analytical part. And then to develop thresholds for reporting, identification, and qualification of leachables, and then provide guidance on safety assessment of leachables. So this is the non-clinical part of the guideline, which is also a huge one. And in scope of the guideline are chemical, biological, and biotech products, including drug-device combination drug products, meaning integral drug-device combinations. So where the device is in contact with the drug product over usually over a longer time period. Medical devices as such are not in scope, as these are defined by the pertinent ISO guidance. Next slide, please. So in the concept paper, it's already written that the guideline aims on to provide a proper definition of leachables and extractables, and to describe the design of a ENL control strategy, which is based on science and also on risk-based principles. Risk assessment has to be done or is part of the process of the component selection, especially the components of container closure, or also the components of manufacturing equipment. We include everything here. We don't exclude, for example, rubber stoppers like the current EMA guidance. So risk assessment is an important part of the guideline, and also the lifecycle management is described for container closure systems, manufacturing systems, and the drug delivery components. The aim is to define harmonized thresholds for qualifying leachables in the context of route of administration, drug indication, and patient exposure, meaning duration of treatment, and to establish limits that are relevant to the route of administration and the treatment duration, and also to define in which way prior knowledge can be used to determine the need for further safety studies. But I think this was my last slide now. Can we? Yeah. So thank you for your attention. I don't have a Slido prepared, sorry for that, but I'm here to take any of your questions you may have.
Thank you, Katrien. And I think that was a very nice complimentary presentation by both you and Claudia on, well, some of the everything going on extractables and leachables at the moment, some referral to specific cases, but also an overview of the guideline revision. That leaves us for with some room for questions that you may have. Before going to the room or to the WebEx, I know we have received one via the WebEx, which is because it was sent in a different way, not visible to all of you, but it has to do with the guideline revision. And it's from Sophie Dufay, and it asks, as the ICH guideline is under writing or under revision, do you have any info on how it will align with the EU guideline, but also the FDA one, that is really setting a strong background on extractables and leachables evaluation? Katrien, or want to?
I don't know, Claudia, do you want to say something about that?
Well, so I think the first point is that because ICH is, there is FDA there is EDQM, there is EMA, and all the other regulatory bodies, the FDA requirements are for sure taken into account when preparing ICH Q3, right, Catherine? So
Yes.
yeah,
We have
and
we have
there
a
is,
lot of discussions.
yeah, so and industry and so on. So in terms of the requirements the FDA have put through, these are considered together with whatever we have in the different territories. In terms of what we expect for MAAs, marketing authorization application. So I think Catherine gave a really good description of the expectation. But currently there is, and please Catherine correct me if I'm wrong, but there is quite a bit of flexibility in the sense that if an applicant comes with an extractable leachable study and specification that meets the FDA guidance requirement, as long as the risk assessment is sound and is well justified, and it also is in line with our plastic guidance, plastic contact material guidance, then that is acceptable and is evaluated in its merit. Is that right?
Yeah, I can confirm that. Yeah, it's it's in the end, it's a scientific approach that has to be followed and justified, and it needs to be demonstrated that the material is really compatible with the drug product solution and does not, or the component, and does not represent any risk for any quality risk, and in the end safety or efficacy risk. But I may also add that the problem, or it's may not be the problem, but the EMA plastic guideline is does not have rubber stoppers in scope, and rubber stoppers are common source of leachables in the drug product, in the final drug product, at least if they are not Teflon coated. But these have to be addressed, and even though EMA guideline doesn't have those in scope, the principles described in the guideline can be applied. So, but in the end, it's a scientific decision, and
All right, thank you. Looking around at the room, are there any other questions on extractables and leachables at the moment? Or online? Please put them in the chat if you have questions. Or raise your hand. I know it's been a long day full of information. And the presentation must have been clear. I see no further questions.
Maybe exhaustive rather. It's quite detailed.
But if you do come up with questions afterwards, also don't hesitate to reach out to us, because we'll be happy to answer them. Okay. Yeah. With no further questions for now, I think that closes this session. So Claudia and Katrien, thanks again.